um, complex versus non-complex sequences. And what do I mean by that? That's a question of the class. What do I mean by that?
The complex sequence has a random sequence.
Has more random.
It's like a random sequence.
Right, but why would this information help you? You correct as to what's the difference between complex and non-complex, and why does this information help you?
Non-complex sequences are most likely in the intron.
That's correct. So the non-complex sequences are found here, and the complex ones are found here. So again, this is all software-based. What they do is they do a statistical analysis of what does the sequence look like, whether it's complex, not complex. And finally, and there's probably even more than they do this, so they also do cross-species homology. And if you want, you can go on top of it. You might as well take into account all cDNAs known. Doesn't hurt if you know you have a cDNA that correlates to this one, and this one, but you don't know anything about this one. You might as well just make a mark that, yes, I know 4 of these. I know 4 of these follow that they're found in cDNA. Okay, so right, you take as much information as you can. And as I said, one of the other things I told you could do, you could look at you could translate in all 6 frames, top and bottom strand, and compare the amino acids to every protein known in the database. And you could say, well, maybe I found this sequence here because it has some bits of homology to a protein. So there's a small segment not predicted by splice acceptor, not splice donor, not predicted by complexity, but predicted because in this little stretch there are 5 amino acids in a row, which are 5 amino acids identical to some protein that I've never heard of. So you could do it by what's called it's called T-blast analysis, where you're translating the DNA in all 6 frames. It's T-blast X. You translate the DNA in all 6 frames, and you ask, does it have any protein homology? Right, so there are many sophisticated programs to get you this information, and there are probably more I'm leaving out. So let's suffice it to say now I have 1, 2, 3, 4, 5, 6, 7, 1, 2, 3, 1, 2, 3, 4, 5, 6, 7 regions I'm interested in. I'm going to draw those again down below. 1, 2, 3. Remember, 4 was predicted by T-blast X. 5, 6, 7. These were predicted by cDNAs, found in cDNAs. Right? And this one predicted by splice acceptor, splice donor. So you don't know, you have 7 exons here. And these are candidate exons. I don't know if they're true or not. So what Affymatrix did was they scanned the entire genome and they said, well, we want to know whether any one of these pieces are in your mRNA. We don't want to lose any bits of hybridization in your mRNA. So what they did was they figured out a technology to bind single-stranded oligonucleotides to chips. So what they did was they bound 18 base pair nucleotides that overlapped. So for example, let's say you had a 100 base pair stretch of exon 1. They would do an 18 mer, bottom strand oligo. Why bottom strand oligo? Because what? Which means what? Why would we need the bottom strand oligo? We're looking for mRNA expression, which is a top strand sequence. So bottom strand oligos that overlapped, and they're all 18-mers. So they came up with, let's say, 6 oligos. They're all bottom strand. Oligos. So they fit nicely into one compartment of one chip. They have, so this is exon 1, and this column here is oligo 1. This compartment here is oligo 2. This compartment here is oligo 3, etc. I'll have to draw more spaces. 4, 5, 6. So just 1, 6, 3, 5. Can't see anything. 2, 4. 6 oligos, all bottom strand oligos, all in one place in the chip. All bind relatively to exon 1. So now you're actually scanning it very carefully. They can ask, if I'm really seeing exon 1, all 6 oligos should anneal to your mRNA, not just one of them. So you can score for exon 1 by virtue of all 6 oligos annealing. In their mechanism, by the way, when an RNA binds to the chip, they are not revealing fluorescence, but a change in resistance. So they have the chips under electrical charge with current going through the chips. And when an RNA binds to an oligo, the electrical current in this little place here changes. And they can measure how much resistance it changes. So they're using electrical currents to monitor whether RNA has bound to that oligo. They were one step more clever than this, because they still feared you would get some garbage, and you actually do get some garbage. And more importantly, we still haven't fully solved the homologous sequences. What do we do about sequences which only differentiate by 1 or 2 nucleotides? We still haven't solved that. Yes? So, let's say, for example, I mean, I'm not a physicist, but let's say, for example, you got 1 amp of current going through each of these chambers, each of these little wells. Now the resistance is going to, 1 amp of current going through, you add RNA, now maybe this one goes down to 0.5 amp of current can go through. So you can measure that difference. Yeah, you know, they don't tell anybody their technology. They just say it in crude ways because they don't want to give out secrets. Yes?
So the 6-column thing, what is that? Is that like a
6?
gel or?
No, it's a chip.
Okay.
It's actually now it's an electric. It's basically an electrical chip of some sort where they can bind oligos to it, to that chip, and they can put a current through the chip. So this is one chamber of a chip, and in fact these chips have 18,000 little one of these units in it.
So once they put the little unit in there with the oligos, do they then sort of bathe it again in RNA?
That's right. You bathe it in RNA. In fact, they do a little bit something different. They actually purposely cut up the RNA. And you could also use cDNA as well. You could use it with. So let me just finish one more thing before we go on. So you can do this for all the exons. You can do this for all the exons. That's exon 1. You can do exon 2, 3, 4, 5, you have a lot of space on this chip. You have at least 18,000 units. 5, 6, 7. It was 7, yeah. And now you can ask, if I really have mRNA expression for a particular gene, I should have many of those exons for a given gene. So for example, if I only get exon 1 hybridizing, it's unlikely I'm actually looking at the gene. I would expect some fraction of these exons to be in before I called it an expressed gene. I'm no longer asking for one exon, I'm asking, and this comes about through sophisticated calling programs. Like you say, do I need 1 and 7 in? Do I need 1 and 6? Do I need 1, 5 and 6 in before I call it an expressed gene? But now you can get very sophisticated information as to which exons are expressed, and you don't score for your gene being expressed unless, for example, you have 2 or 3 or 4 of them hybridized. Yeah.
I think you got into this of knowing which exons specifically are expressed, but why the 18 base pair? Like, why do you know which oligo was bound to as opposed to the whole exon?
Well, this gives you specificity. For example, if you only got oligo 1 binding and not oligo 6, more likely to be that you're getting cross hybridization with something else.
As opposed to if you had oligos 1, 2, and 3 or the...
Right, even 1, 2, and 3 you could have cross hybridization, but it's unlikely you're going to get 1, 2, 3, and 6 and it not be real. Yeah.
So for 2, 3, and 5, what does that mean, the red and the blue? Does it mean that?
No, it's just different oligos. I just color-coded them for you.
Oh, so it's not like, so it's orange measuring, so it's.
No, this is nothing to do. I just color-coded different oligos.
But, so does the chip just, so each chip with each oligo is measured to the like different?
Yes. Yes. Each little unit within a unit is measured in different terms. They're very sophisticated chips. Remember, these are sort of like your system software people that make your chips for computers. They're doing the same thing, but with DNA onto little units.
So you wouldn't include any intron sequences because of the predictions by the software, right?
Well, there's only 18,000 places on the chip. You have to make some decisions along the way. It's just too much information, and as I told you in one of the previous lectures, you're full of repetitive DNA in the genome. That's going to be hard to score against. And in fact, one of the things you do is you get rid of repetitive sequences that are in your exon so you don't have to bother to get false positives. If there happens to be anything that's not unique in your exons, you would never put them on one of these units. Yeah?
What would be the interpretation of like a whole exon being convincingly real but not any other part of that gene that you're scanning for? I mean, would that be like, would you think that's an exon for another gene?
Yes, it would be the simplest explanation would be an exon of another gene. But I'll get into a moment how they've gone one step further. Yep.
So if you got RNA binding at oligon receptor 4, something like that, would that mean that 4 wasn't a real exon?
No, you mean here?
Yeah.
So you got binding? Yes. For example, that you would say that at least for that RNA you looked at, in that particular cell you looked at, 4 is not real. For example, maybe 4 only is expressed in one subset of kidney cells, but you did breast tissue. You don't find it there. Yeah.
Or are we no longer doing thousands of genes at a time?
Well, we had 18,000 places, and this is just 7.
Okay.
Yeah.
Okay, so each of those are chipped with the 1, 2, 3, 4, 5, 6, 7.
Each of these represent one unit in their chip. That's right.
Okay, so each of them represents a unit of the chip. And in these chips, there are oligos.
Right.
And there are oligos that separate for each parts of each exon.
Within each exon, yes.
Each exon.
Yeah.
So we can tell by using this whether an exon is real by whether it all binds or some of it binds.
That's right. Right. So They did actually, so I sort of lied because I have to take it slowly with you guys. Yeah.
I was saying, like, what is it about the oligos binding to the exons that make it real? Like, why would we expect oligos to bind to
So
real
there's a
exons?
perfect segue. So why I lied to you is because there's actually a real advantage of using 18 bases. I'm going to see if I'm going to start probing the class and seeing where you are. So what is the advantage of, what is 18 bases useful for? Complexity, specificity, anything else you could imagine that might be useful for doing along the lines of, he said, what would be good about using oligos?
Well, you use them for your primers in PCR because they're specific enough that they can bind to a sequence, yet they're also complex enough that they won't bind to.
So what offers that specificity? We went through the probability.
Probability that it will bind into 1 to the 4th to 18th power.
Right. So anything,
It occurs once in the genome.
it occurs once. Let me tell you one more thing which maybe I haven't taught you. I have a feeling I didn't teach you. When you do a PCR reaction, you have a second benefit from using primers. You use primers, blah, 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 blah. These have to be a minimum of 18 bases. Remind me how PCR works. We denature the DNA, then we add 95, then what do? At what temperature? 50s. And then we raise the temperature, 72. Notice this middle part of anneal at 50s. Can we anneal at 37? Can we anneal at 75? Why 50s? Why 50s? Yes? So his answer is the melting point of the primers. Does anyone have another way of stating that?
So that the primers have enough heat so that they can bind, but you also can have your DNA bind by it.
Right. So actually the DNA does bind back, but you are so saturating in oligo concentration that the oligos beat the DNA back to reannealing. And that only happens at a certain temperature, which is provided by the length of the oligo sequence. So for example, a 15 mer will never bind in the 50s. 50 degrees C, it will never bind. Because it doesn't have enough energy, it doesn't have enough stability at 50 degrees to maintain the hydrogen bonding. You need at least an 18 mer to bind at 55 degrees or so. That is the minimum. Otherwise, if you're a little bit lower than 18 nucleotides, you won't bind. So there are these balances, and 18 turns out, 15 was what we needed for complexity, 18 is what we need for efficient binding at what we call melting temperatures. If you have a 22 mer, you can anneal up to 65 degrees or even higher. You can actually, with even a very long one, let's say 25 mer, you can probably anneal at 72 degrees C. And actually, I think, maybe Rio, you can catch me if I'm wrong. I think it's called touchdown PCR. Is that right? Where you just go 95 to 72, and at 72 you have enough temperature to anneal, and you can just go. So you just cycle between 95 and 72 because your primer can already anneal because it's got enough length. So you don't actually have to go through that annealing phase. You can just cycle between 95 and 72 if your oligo is long enough. It's not as efficient as regular standard PCR where you do a separate annealing phase, but it works. Okay, so now let's go back to this. Why 18? Yeah.
Don't you have to take
Don't you have to take their melting points into account for?
When you figure out the annealing temperature?
Yes, so you can calculate them individually for every set of oligos, and again, like I said, I sort of lie, I tell you it's 18, they're not all 18, some are probably 17, some are probably 19, but they're roughly around 18. I lie. But there's another good reason why 18 MERD might be useful. It has to do with what I just said. What did I just say about why we have to be at 18?
So pretty much, we have to be at 18 to still increase the chance of it binding.
It has to, right, so melting temperatures for DNA, RNA, DNA, all pretty much have to happen in order for it to be efficient around 55 degrees. So, what is the other useful thing about an 18 MERD? What?
That you can make, well, I think someone said this already, but you can make the PCR, or not the PCR of the specific exon 1, 2, 3, or 4, because each of them.
So this has nothing to do with PCR about why. Remember, none of this so far is PCR based. This is only annealing. All we're doing is annealing and recording changes in current through these cells. No other ideas? How about somebody who's done an Affymatrix chip? Anyone done an Affymatrix chip? Yeah?
Can you use 18 because of the current supply chip?
No, it's not for that reason.
Maybe for the overlap, to have enough excess nucleotides to attach to the previous and the after.
It's not for overlap. I'm going to stop you guys in a moment, but yeah?
Couldn't you also use it to add, like make a complementary DNA molecule? Like add DNA polymerase to make a complementary?
No, this has nothing to do with polymerization. It's simply to do with annealing. Last time. Say it again? Well, but that's what I told you already. So it anneals at 55 degrees. So the answer is, so it turns out, as I said, I lied. Each exon, each chip, each coordinate has actually a partner coordinate to it. So for example, so if I go back and I do these units, 1, 2, 3, 4, 5, 6, 7, what they've done is something very sophisticated. They've taken each of those chips, each of those oligos, and in a very neighboring chip, the exact neighbor to it, they've introduced a point mutation in the middle. The point mutation effectively blocks hybridization. Thus, it gives you something to measure backward. So that if it just happens to be anomalous binding for whatever reason to just a fraction of that 18 mer, you now will measure it with these. So what they've done with the 18 nucleotides is they've introduced a point mutation dead center, creates 9 and 9 as the minimum amount of hybridization. So it's very, very weak binding. And it measures background. So actually, each one of these chips here has a background detector oligo for it. This is a very sophisticated chip. Not only do you microarray, you check each 18 base pairs, they overlap, but now you actually check what is the likelihood that is noise by using a point mutation in the middle of the oligo.
Sorry, the background for the oligos that are being caught in that one well?
Yes.
Okay.
Right. So it's these same oligos here. Those exact one are in a neighboring compartment with a single mutation within the oligo so that it can measure noise.
And by noise, meaning like, so basically.
Just the 9 binding or just the 9 binding
So
here.
we can see if it's just that all of it binds perfectly fine, or if it's just that
Right.
most of them bind, but for some strange reason, 3 doesn't bind, or half of 3 doesn't bind.
Right.
or
So
half
for
of 5
example, you could have binding here and here, which might look like it detects signal. Right? You could get some amplification, but because you're missing this one nucleotide, you're going to be a lot less binding efficiency than if you were to bind all 18 in a row. And it will measure that as background.
So this is just giving us like false positives.
So this measures the rate of false
And
positives.
it takes it out of the
And,
total.
well, you would have to subtract it. So, in fact, so you see, it's very sophisticated. Going back here, you not only just measure the degree of current change in a chip, you do it relative to a control, and you relative to exons. And this is thought to be quite good technology to be nearly rock solid. You've gotten your gene of interest, and remember, this will pick out also genes with single base. You can now design chips where they have single bases different when you have orthologous genes, genes that are, or paralogous genes, things that are very, very similar in genome. You can design those oligos to see that mismatch if you want to. Yes, Sonny?
So are you doing it again with the fragmentation, or you're doing it
Simultaneously. Every compartment has a control compartment associated with it. So you actually never measure this alone. They always subtract the negative control from the readout experiment. Always. So each one is associated with a negative control. Of course, that takes away our 18,000 compartment, 18,000 units. So, you know, now you have, for this particular gene, so you actually only get maximum 9,000, and of 9,000, if you have 6, 7, 8 exons, you're now down to, let's say, 1,000 genes if you're looking at it. But you can actually, they're willing, the company will array any oligos for you. So you can give them a list of oligos, and they array them. They make the oligos, and they array them, and people make affirmative chips for all kinds of different reasons. And there are all kinds of selective chips that are being made. Yeah?
Like you just mentioned, this is happening with the presence of orthologous genes as well. It's the whole mRNA from a cell that are going over. How do you know that exon 1 was bound from that predicted gene and that predicted exon and not some other mRNA? Like, how do you know that exon 1 and exon 3 were bound by the same mRNA and not some of one was bound by one gene and 3 was bound by another?
Because again, you would say that since you have some idea, let's say this has, and of these, this has the only poly A, you would call that background. It's unlikely that you could get 1 and 3 without 7. Hypothetically speaking. I mean, and again, they rig the chips that way. They rig them so that you can call them out. So I don't know if you can tell, there's a huge bioinformatics going on when we do microarrays nowadays. It's not like the old days where you're just doing a hybridization looking at fluorescence. Now these are sophisticated call programs where we decide what's a good hybridization or not. Yeah.
The assumption behind this is that you've already sequenced either the gene or the genome, right?
So this happened right after all the genome was sequenced. That's correct. That's correct. That's why Informatics took off. Right after the mouse genome and human genomes were sequenced, they landed the entire share of the market. Yep.
Wait, just going back to what you said up there, and I think what he said made sense. Are these oligos coming from an mRNA? Like we're splitting up the mRNA to see if it will attach to these?
The oligos are usually derived from genomic DNA. They're based on, as I told you, one of the parameters might be cDNA, would be based on, but they're coming from genomic sequences. Remember, you have the entire 3.9 billion nucleotides in a database. They simply design oligos to the bottom strand and make them. 18, whatever, 100,000 oligos they'll make.
So we have the cDNA and we're splitting it up to see if
You're not
it
splitting it up. These are synthesized nucleotide oligos. Oligos are generated by an oligosynthesizer. You have a column, you add a base, you add another base, you add another base. These are synthesized in the laboratory, these oligos. They're not cut from anywhere. They're derived from many places. Right, I said you could go to many sophisticated ways to pick out what in genomic DNA you might be interested in, including the all-known cDNAs. that map onto these regions. There are many sophisticated ways you would go about doing this. And again, as I said, you would come up with, as I showed you here, a heat map based on sophisticated algorithms by which you decided, I'm only going to say a gene is correct if I have all the exons there, or 3 out of 4, or whatever. Yep.
So the ultimate goal of like both these techniques is to measure RNA expression, right?
Right.
But so what would the advantage be for using this as opposed to like the reverse PCR where you're basically doing the same thing? Is that just more time intensive or something?
qPCR is just for one gene at a time. This you screen thousands of genes at a time. However, you've reminded me of something that has the potential to obsolete all this, although maybe not just yet. And does anyone know what that is? I mentioned it a little bit before. Does anyone know? So it touches upon what you said is, well, here we're doing an inferred count based on hybridization. Does anyone have any other ideas how we might get at RNA levels? What's a simpler way to do this? Faster, probably not cheaper. Yes, Dana? And what way is that?
I have no idea.
Okay. How would you count them directly?
Like gene by gene, like a qPCR.
So qPCR is doing it gene by gene. I mentioned it once before. All right.
I remember you mentioned a company called Nanopore.
Well, that gets to the point I'm saying, which is you can do what's called transcriptome sequencing, where now you're taking all the mRNA in a cell, and you're making first strand cDNA, and you can even amplify it by PCR. And you just sequence everything. And what that looks like is, so for now, let's go back to our 7 exon gene. Okay. So that's our 7 exon gene. And now when you do this transcriptome sequencing, you get these little fragments of sequence, 50 to 100 nucleotides. And you get these little fragment reads. And you have to use bioinformatics to align them. This is just one gene, and you do an alignment. You actually do an alignment of all the sequences to everything in the genome. Takes some doing, but again, there's some sophisticated software to do it. You can allow for one mismatch. You can do all kinds of things because again, remember, nothing is perfect. DNA sequencing reads will have mismatches. You can say, I'll allow everything with one mismatch. So you can say where that mismatch is, at one end or the other end or in the middle. You can ask anything. And what you do is you get an alignment of all the sequencing reads, and you can map them onto the exon. So this is actually important you understand this, and I'm even not going to go through the next lecture, the next part of the lecture I want to go through, so that we understand this. So these are sequencing reads. What we've done is we've made mRNA. We've made first strand cDNA. We have now DNA as a template. There's mechanisms by which you can anneal oligos to that cDNA. Once you've annealed oligos to that cDNA, you can make a lot of it. Theoretically, you you haven't changed the stoichiometry of any given mRNA. That if you've made mRNA and you've made it cDNA and you amplify it, hopefully the ones that were low expressors are still low after PCR and high expressors are still high after PCR, but there's no guarantee of that. And then this is sequence analysis of those cDNAs. And you only get little runs of cDNA. You only get these little stretches of 50 nucleotides. And if you now map those 50 nucleotides onto the genome, you get these patterns. And so you can see here I have a lot of little sequencing runs under exon 6, and I have a lot of them under 7. I have 2 under exon 4. That's it. But notice they fit nicely under there. I have less for 1, 2, and 3. That means all my exons are expressed. And it's a question of whether this extra expression here is real or not. It could just be a reflection of how the RNA polymerase is going through the RNA, and it's not a reflection that this actually means there's more cDNA. Probably it means there's this level of cDNA in the cell, probably the average of all the hits. And it means that some rare transcripts are picking up this exon. A few rare ones are picking it up. Yep?
So if you've got that many fragments under 2 of those exons, and it was real, that would mean that those exons were in that many more transcripts from this gene.
That's right. That's right. But there is some unusual things happening from transcriptome sequencing, and it's not as equal as you might want to see it. And I think at this point it's unclear why you don't get equal distribution. But what you can say is that even though you don't get equal distribution, you can say that the ones that are super low are really low. Right? And the ones that are really, right, you can see here 1, 2, 3, 5, 6, and 7 all roughly have the same amount, although 6 and 7 have slightly more. For the time being, we just ignore that. There may be information there, but we ignore that. Yeah?
And the number 4 is the control?
No, number 4 was this hypothetical exon. Remember, that was, I called it early on. I called it, I said, this came out by protein homology. Not by splice donor, splice acceptor, not by ever finding it in the cDNA. I only found it from a very sophisticated software. That's how I found it, and voila, when I do the sequencing, I find it. So in fact, transcriptome sequencing should kill microarrays. It should completely kill it. It's still not popular enough. It's still a little bit expensive. It's roughly about $1,000 every time you do it. Microarray might be in the $200. Once the chips are made, it's maybe $100 to run a chip. So when you're not rich, you do the microarray. When you're rich, you do the transcriptome. Because transcriptome, as you can see, is beautiful data. It tells you exactly. It really does map up. I mean, like this. And in fact, what's really alarming, and this is the part that really I find amazing, you see databases have exons like this. And what you find is that when you look at the transcriptome data, you get stuff like this. So there is no exon called for this in the genome browsers. But when you do the transcriptome, you get something that looks like there's RNA being made here. Either it's artifacts of mismatching during the alignment protocols, or there's a real exon there that just no one identified. And this happens much more than you would expect it to happen. Happens a lot. So again, the programs are terrible at calling what might be hidden exons. Yep.
Is this a contiguous sequence of the genomic DNA?
Yes, you would basically have the entire genome on a browser. I mean, I can look through all of one chromosome at all the hits along the chromosome, and I can scan it.
But what we're looking at is,
Yeah,
you're looking at
this is 4 exons of a given gene,
from 5 times.
from 5' to 3'. Yeah.
Do you use a software before you start this to predict it as well, or do you just go
So
straight
for
to-
transcriptome analysis, there's nothing to predict. It's just you sequence and you align. That's all there is. That's why it's so powerful, because you don't make any assumptions and you get to see the raw ingredient. Yep.
Does the transcriptome also pick up microRNAs as well? Or is it
Yes, the transcriptome picks up microRNAs.
I understand you get these deep representing exons, but again, if you have all of the mRNA from a cell at once, how do you know which exons correspond to which genes? And like, do you have a lot of them?
You align them. You align them. So you get 60 million reads simultaneously, and you align all 60 million sequencing reactions to the genome simultaneously.
Ha